here. I'm just going to go ahead and hit record. Okay, great. So just by way of introduction, my name is Fred Colling. I'm the director of the single cell genomics and spatial genomics shared resource here at Dartmouth College up in New Hampshire. Our group has been doing single cells since about towards the end of 2016, and we've been doing spatial work since 2019, towards the end of 2019. So we have a lot of experience in that space. We are part of a cancer center, so a lot of our focus is on human and mouse samples with a pretty heavy immunology focus, but we also are the core facility for the college as well. So we have people that are trying to do corals and axolotls and other sort of less common organisms as well. And so we have a little bit of experience getting that to work or struggling to get that to work, depending on the situation. So we can speak to a little bit about that as well. But I think really, the goal of this discussion is really to be a discussion. I mean, I don't really plan on sort of talking at you guys about things like this. I'm really interested to know sort of where you all are at and what sort of questions you have about single cell and the challenges that you guys face in your own facilities. So I don't know if anybody wants to jump in with anything that immediately comes to mind. Hopefully we can start a helpful conversation here.
All right, someone's got to start. I guess I'll try.
Thanks, Karen.
I'm Karen Abramson. I manage a genomics research lab at Duke. We primarily use 10x Genomics for our spatial and our single cell. And we have been experiencing for some time now, at least 6 months, that our capture rates are going down. We try to target 10,000 cells and we get maybe 5,000. And we're loading it according to their protocol and their tables. And we have gone round and round and round and round and round with tech support. We have ruled out the instrument. We thought it was the Chromium X because it kind of happened when we switch over to that. But we did a side-by-side between the Chromium controller and the Chromium X, got the same results. They thought maybe it was our cell counting, but we've been doing the same exact cell counting since 2016. And we did a whole bunch of testing with that and ruled that out. That was not it. We have had tech support watch our techs load the chips. They say they're loading them fine. So I am very curious and just dying to know if anyone else is experiencing these problems, because of course they tell us it's just us.
And so this hasn't been corrected. This is the current state of affairs.
Yes.
I'll say from our perspective, I don't think we've seen that issue, right? There's always the cases with some strange samples, right, that are maybe hard to accurately count, or the quality might not be great, where the capture rate is lower than you're targeting. I don't think I've seen that on our end, for like PBMCs where we're trying to capture 10,000. I don't think we've really had any issues there.
Yeah, we very rarely run something as simple as PBMCs. I mean, it's usually more from primary tissues, but we do see it with all sorts of different sample types. So for now, we've just taken to loading more. And sometimes we end up with too many. The next step is 10x is coming on site to do another side-by-side test of them running and us running. And we'll see what that shows. But it's just strange that it happened so suddenly, and we've ruled out all these things. And they're very evasive when I ask if anything has changed on their side. Has anything changed with your manufacturing, your sourcing of materials? They don't answer those questions. But I have noticed an increase in other notifications coming from 10x very recently of this buffer's bad, this reagent's bad, we need to replace this, we need to replace that. So it really just does make you wonder.
Hey there, I'm Kevin Childs at Michigan State University. We aren't doing, we have a Chromium. Unfortunately, there aren't lots of folks here doing single cell work, but I believe you. and I suspect it's 10x. We have occasionally very similar stories with Illumina, where we see issues and they say it's not us, it's not us. Like we've had like at least 3 times where these back and forths go on for 6 months or a year, and then later they say, well, it was us. So what we wind up doing is, like, whenever we begin to see an issue repeat itself, we're very closely watching lot numbers and, trying to record everything so maybe we can find the pattern, because they don't seem, despite all the resources, capable or willing to kind of dig in at that level. So maybe that's one thing that you can start to do, is really pay attention to lot numbers.
Yes, we do track all of that. We're tracking everything we can possibly think of. So we'll see what this next test shows. But in the meantime, just very much cautioning customers that you might not get what you're targeting. There are a lot of variables, a lot of reasons to why you might not. So just trying to manage expectations.
Yeah, I think that's the tricky part, right? Is there's so many, and probably from Tag's perspective too. I mean, maybe they see that people are under capturing, but there are so many variables that go into it, right? It's different cell types, it's different cell counters. It's really hard to nail down, for them to parse out, I think, what could potentially be user or other instrument error versus what's happening on their platform. Not to say that it couldn't be something happening on the 10x side. I wouldn't be surprised if it was, but it's probably a little hard to pin that down with so many other variables in the process.
Right.
But
have you only been running the standard throughput, or have you tried the high throughput too and have been having that issue?
We also had the issue with the high throughput. We've only run the high throughput literally twice.
Okay.
But yes, we also had the issue there. They were actually the first customer who was very much like, hey, we've run a bunch of these things before and they were fine. Why is it all of a sudden not fine? So
it's interesting.
Yeah. I have a question for you, Karen. So I was wondering what type of cell counter you have.
We use the Ceilometer K2 from Nexcelom, which I think at one point 10x was recommending in their protocols, and now they recommend a different one from Nexcelom, a more automated one. The issue with that one is it requires a large volume, and most of our customers are coming to us just barely grasping enough cells to get what they want. So that's why we haven't adopted that one.
I have the auto one and definitely don't adopt it.
Really?
Yeah, I was like,
Really?
I ran into so many problems with it. I've also had it repaired by them now 3 times, and I've owned it for about 1.5 years.
Oh no.
Yeah, and so unfortunately the problem with them fixing any problems is I have to ship it to them, and sometimes they don't have an instrument to ship back to me, so I'm without a cell counter for a period of time.
And that company has changed hands too.
Yes.
Because it was Nexcelom, and then they were bought by Perk and Elmer, and now they're like Revity or something.
Correct. Yeah, and I just finally got so 10x told us you should buy this cell counter, so we did, because we had the grant money at that time to do it. So we're like, of course, you recommend this, even better. And so we bought it, and we've had the 3 changeovers. I just got mine back finally, but have refused to ship out their other one until I can test it quite a few times to make sure I'm not going to have the same problems again. Because it was like, I'd put a sample in, it was telling me like, oh, can't see the sample, and would just like abort itself. And I was like,
no.
this is not good. I need to move fast whenever I'm like running these samples. But I also just had that instrument. I'm at the University of Oklahoma Health Science Center. I had it delivered to the other side of campus, which is about 15 minutes from here. They happened to call me saying, hey, I think your instrument got delivered here, which wasn't even part of the university at all. It's like a separate entity on the other side of campus that I got delivered to. And so I was like, I don't think that that's my instrument. Are you sure? And so I called Revity, the rep, and he was like, oh yeah, they must have just shipped it there. Are you able to go get it? And I was
sure, why not? I'll just throw it in the back of my car.
And that's what I had to do. I was like, I have no other choice. Like, it's 15-minute drive. I'm not taking a cart and taking my cart across campus to like get this instrument. Yeah, but we've had
crazy.
nothing but problems with it. But that's why I don't want to. I have your instrument right now, is what they sent me to cover during that duration of time. So I have been waiting because I have a bunch of samples coming next week. So I'm not gonna send out their instrument until I can verify that the new one is actually working.
And that's
Very
that's
good
the
to know.
Celica
That's-
MX.
Yeah, that's it, right? The Celica?
Yeah.
What do you use in your lab, Fred?
We use a K2 also. I love it. I mean, I really like the K2, but we were going to get a Celica, but there's actually a lab down the hall that's getting a Luna. It's like an FX6 or something like that. It has like an 8-chamber slide option for higher throughput. So I think we might see how that plays out. Well, now definitely after hearing Kylie's
Yes.
experience, we're totally going to try that before buying a
Yeah.
Celica.
Yeah.
Yeah.
And the K2,
I would recommend
K-
you. Sorry,
Go ahead.
we just purchased the FX7 from Luna, and I'm really happy with that. And the advantage is that you can do like a demo, so they will ship you the instrument for a week, and you can test it with all the samples, and you can run up to 8 samples in one slide, so it's very convenient. Yeah,
That's
we
great.
like it.
Yeah, I've really liked the K2, but I mean, for a couple of projects, I mean, for multiome, sometimes we're doing a handful of samples at a time for multiome, and you just end up counting nuclei a lot, so that can be hard to do on the K2. And then definitely for flex, you've got 10, 16 samples or something like that on the bench that need to be counted 3 or 4 times between doing the hive and loading the machine. I mean, doing that with a single shot counter is just, it's a
Yeah.
nightmare.
Yeah, I think we
Yeah.
do need to eventually invest in something more high throughput. So it is very good to know that there are all those issues with this Celica.
See, they'll fix that in the future, but as of right now, it is not worth having it.
And it's
Because
expensive. It's very
yes, it
expensive
is.
machine,
And
too.
it costs a lot to keep it and for the service warranty for it. And of course I have to keep the service warranty. There's no way that I trust the instrument to not need to get repaired.
Yeah.
No
So
kidding.
yeah, yeah, it was
Yeah.
actually one of those things. Like my director told me, he's like, "Do you think we have to get it this year?" And I was like, "Unless you want a paperweight, like, yeah, we need to pay for the service warranty, or it really is going to become a paperweight in the next couple months when I'm having another problem with it." We'll see.
Mhm.
Does
Yeah.
anyone in here have a Connect from 10X? No? Good. Okay.
Yeah.
Do you?
Yes.
Do you like it?
Unfortunately.
You don't like it.
I just run into a lot of problems. I've gotten a new instrument already, like a full new instrument after it being repaired 3 times. So I'm biased in it. I also see that, like, as much as it's convenient, if you don't have the time to have a technician actually do everything by hand, it's nice. So then you can just press play and walk away. But what I'm getting out on the other end is showing that it's a robot doing it. Like, when it's doing its cleanups, it's leaving ethanol, like it's leaving things that, like, then I have to go back then and do another cleanup on it, ruin basically that library. And it's easier for me to just go back to the CDNA and make a whole new library myself. So, and we've just had so many problems, like technical wise, that like I don't trust it to press play and walk away because I've had it error mid run. So it has been halfway through its CDNA production and just had a big red screen that had abort. And I then couldn't save the samples. So like it couldn't even tell me at like what step it was. It had already passed like gem creation. It was in some component, but it couldn't tell me at what step they were. So I couldn't even try to save the sample and like continue to run it by hand. So I'm not the biggest fan of it. I'll use it if the PI wants to use it, because some PIs have been very adamant with me that they want the robot to do it because then it's consistent for every single one of their samples and it's not a person doing it. But they usually regret saying that to me a couple months later after they've like sequenced and done all of that. It's convenient for them, and then I can train them just how to load the robot, put all the things on, and then they can press play and walk away, and they don't need to wait on me to get those samples done. It also worked out very well with like a PI. He won't be able to give me samples till like 10 o'clock at night. And I was like, not staying here at 10 o'clock at night. So I was able to train him how to use the Connect. So then I don't have to be here at 10 o'clock at night for him to be able to run that instrument. So it has some conveniences like that, but not
But
worth
not enough to be worth the cost of it.
No. Nope. Yeah, because we deal with a lot of like aging stuff here, and so like all my samples are as precious can be, because it can be a human tissue or even like a mouse tissue that's years and years old, and it's the only portion of tissue that they have from that disease state, and then I lose it in the instrument. So when it comes to like super precious things, I tried really hard to convince the PI to have me do it by hand rather than use the robot, because I don't trust it, unfortunately.
To switch directions, we have a minute and 54 seconds left. I don't know if it was worth touching on. Does anybody here get involved with sample prep for single cell in their cores?
Yes.
We do lots of that, but so can speak to that. I don't know if that's something that other folks do and are having trouble with or have questions about, or
Yes,
horror
so
stories to share.
yeah, actually speaking about the K2, so I'm having problems with some scratches in the slides. I don't know if you guys have the same issues. So the slides came with scratches, and so these scratches are being counted as cells. So I need to change the way to discount these scratches. So to do a new assay to discount these scratches, but it's something recurrent. So we order new slides, a new lot. So it came with the same problem. So I wonder if it is someone else has the same problem.
We don't. Do you buy the pre-peeled slides or the ones with the film on them?
Yeah, it is the Nexalon K2.
The slides that he's talking about, so they make two different sets. They make ones that you have that are pre-peeled and don't have the extra plastic on both sides, and then they have ones that have a peel that you have to do on both sides of the slide before you can use it.
Oh no, I need to peel. Yeah, I need to peel both sides of the slide. Yeah. Yeah, I
Those are the
didn't
ones
know.
that we use too, and we haven't seen that issue.
I also
Okay.
haven't had to purchase them in a while because we bought in bulk initially. So
Oh, okay. Yeah.
has the company been told, and have they said anything and given you free
Yeah.
slides at least?
No, what they said was help me to set up a new essay to discount these scratches. So yeah, but it's really bad.
Huh. We'll keep an eye out for that if that's a new lot. The same thing we've probably had ours for a year now because we bought a whole bunch, but
Right.
we'll
Yeah.
keep an eye out for that.
Yeah.
Well, thank you, everybody. Sorry to cut it short, but it looks like we're all going to get kicked out in 6 seconds. So you can find my email anywhere. Hopefully we can all keep chatting.
20 minutes is not enough, man.
Yeah,
Never enough.
Not enough time.
yeah. No, the discussion's just going, and I think this is fantastic. So we have, I'm gonna we can do the recaps that we've done in the rooms, or we can go back and do another 20 minutes. We have 30 minutes left in this session, right, Ken? Yeah.
We have as much time as the group wants, Kevin.
Yeah. So we can go back and reopen the rooms for another 20-minute session.
So Kevin, may I suggest that we go back to the same rooms for 15 minutes and then recap for 15 minutes so that people, when they go and watch the videos in the other rooms, if they watch the videos in the other rooms, at least will have some idea as to what's being talked about, can choose for themselves what's important for them.
So they can just open it up for another 15-minute session. You can stay with where you were at, or you can jump to another session. 15 minutes. All right.
Aaron,
And we're back.
I just wanted to make a comment regarding what Karen was asking earlier about the capture. I had issues with the high throughput kit, the HT kit. I aim for 10,000 cells and I have like 6 samples and I recover roughly 5,000 in all of them. And then the next round, I targeted for 20,000 cells and in all 6 samples, I recovered 8,000 cells. So it was less than half. So I faced that issue with the high throughput kit, not the regular standard 3 prime. So I don't know. I'm right. I have a ticket with the next support at the moment and with troubleshooting. So I have to give them all the details of the experiment. So hopefully we can figure out what's wrong. Yeah.
Hi, I'm Alora. I work for Karen in the molecular genomics core at Duke. I was just wondering what cell type that was, just out of curiosity.
That experiment was with PBMC.
Okay, that's what ours was too, that we had the issue with the HT, which is very odd.
Yeah, because we did like a first cell count using a hemocytometer, like manual counting. So we were like pretty accurate in the cell count. I usually trust the hemocytometer very well. And then we did another count with the automated fluorescence counter that we have. So we had 2 rounds of this counter and we were in the range that we were expecting. So I was pretty confident with my cell count.
Yeah, we've gone back and forth with them on the cell counting so much, and they've confirmed that that's not the issue. So yeah, for us, we were having way lower capture rates. We started loading more, and now it's just all over the board. We're loading like 19,000 cells. We could get anywhere from 4,000 to, we've been seeing like 35,000 somehow, which isn't making sense with their algorithm and what's going on. So we don't know what's happening, but it's really weird. And there's no explanation for us to be getting more cells than we're loading now.
Yeah. And those are PBMC, you said, right?
Not
Oh, you're
always.
getting this with different cell types.
Yeah, we run a lot of different cell types.
But all the testing that we did comparing instruments and
Yeah.
comparing cell counters was on PBMCs.
Mm-hmm.
Took the exact same sample, split it up, ran it on different things.
Yeah. I think sometimes like cell size may have, there is like a limit in the channel, like how the flow goes with cell size. Maybe that's something like a parameter to keep in mind. I don't know how it would affect, but that maybe like some variation may come from cell size and cell rate capture.
Yeah, we've started asking our customers for their average cell size if they know it, so we can change our gating. And that hasn't helped us either. We've still been seeing the same thing.
It's like Trisha has a comment.
I do. I'm Trisha. I'm from the University of Michigan Genomics core. So we have had a very strong correlation that when the cell size gets smaller, then that's when your cell count goes extremely low. So we've had I have a pancreatic customer who comes all the time, and we've had to double her number of cells that she loads. So instead of putting in 20,000, we put in like 40,000. And then she finally gets her 8,000, 5,000 cells, because if we didn't do that, then she's only getting like 2,000. And that was extremely consistent. And we have somebody who's had some tumor samples, like brain tumor samples, that are the same way. That they start recommending just overloading it because it's okay. It can handle it. For whatever reason, those small cells just don't go through.
Have you been seeing stuff on the other end though, in the bioinformatics of it getting doublets and triplets and all of those issues with having to put that many cells in there?
No, it's just for whatever reason, it's like they aren't heavy enough or something's not happening to push those through.
Is this then also true with, I'm sorry, is this also true with nuclei then?
You know what? We don't have that problem as much with nuclei. It's just the like really tiny cells. When your average is like 3 to 4 microns, that's when you're seeing that just not traveling through.
Yeah, we had a lot of trouble with a fly sample with
Yeah.
those kidney cells.
Yep, fly samples do that too.
Yeah, we ended up overloading to get 10,000. I think
And
we
then we
loaded
got-
20,000. We got
and then we
like
got more.
20,000. Yeah.
The thing I
I-
wonder with that too is whether or not, because right, a cell size goes down, just like transcriptome complexity goes down too, right? So it could potentially be just a sensitivity in the chemistry issue, right? Just that less complex cells were not, if there's issues with the enzymes or something like that, that could be impacting how well you're amplifying the transcriptomes from those. So I do wonder if I don't know if you play around with this, not that you're going to recover the lost sensitivity, but if you play around with the cell ranger cell calling and sort of push that out to where you expect it to be, if it says you captured 5,000 cells, you were trying to capture 10, if you do the 4 cells equals 10,000, are those last 5,000 really all junk, or is it kind of like, just low complexity, but actual cells? That might be interesting to look at if you haven't already.
So you're suggesting that if they're low complexity, the algorithm is not calling it as a cell, even though it is.
Right. And if there's an issue with the chemistry, right, that low complexity is lost even more so in the data, right? So they may be getting captured, but you're just not getting good RT efficiency or amplification or something like that. And so they're dropping out at a later stage, which to me seems more likely. I mean, I guess I'm not an expert in microfluidics, but just in terms of like the chip design and things like that, it's not immediately clear to me how the performance on a particular cell size would sort of change with manufacturing lots. I feel like as long as you were still getting similar quality emulsions, consistent droplet sizes, like that should be consistent, but be very easy for enzyme lots to have issues, and it could actually be on that end.
Yeah, no, ours is consistent over like 3 years.
Oh wow.
We were like,
Wow.
yeah, it was that they were like, okay, we've been doing this for 3 years and we still haven't been able to get more than a couple thousand cells out of each sample. So can we up that a little bit? With beautiful knee plots, like there was absolutely nothing that suggested that we should force cells higher or anything. All it did was create more background. Wasn't something that clustered out. But on the other hand, I had a question about like the 35,000 cells. Like, is that like kind of what do those plots look like? Because is that calling background instead of actual cells?
I'd say we've seen both. Sometimes we can figure out that, okay, it's calling a bunch of ambient RNA cells when it shouldn't. But other times, no, it looks really clean. Somehow we got that many cells. So it's consistently inconsistent.
So I
don't know how much time we have or if we still want to talk about 10x, but I'm curious with all the issues with 10x that people have tried other single cell technologies and what they think about those.
We run a little bit of parse. Just like, yeah, very little bit, like not really, if you're billing for all your labor and everything, a lot of the cost savings of that scale sort of go away. It's a lot of work for our core lab. So we've done a couple one-off projects, but not routinely. We have like doing honeycomb a fair bit. The sensitivity is not quite as nice as 10x or even parse. But the workflow is actually pretty straightforward. And we've been using that in cases where like people want to do the flex, people want to do some sort of preservation or whatever, but it's not a mouse or a human sample, or they want longer, they want, what do I say, like real transcript sequence as opposed to just sequencing probes. So we've used it for that, and it works quite nicely. The cost is reasonable. So we've liked honeycomb.
So parse, other than it being more time, how are the results? The results good?
I don't know about you, but we're having a really hard time with yields.
And is that what types of samples?
All kinds of samples. I've done a bunch of mouse kidney. We did some mouse PBMCs, we've done some human stuff.
Mm.
Yeah, but I don't know, like the other problem we have is that a lot of our customers don't have that enough cells to fix that by the time they get to the end, they can target what they were trying to target.
Yeah. Yeah, and the amounts you need for fixation for parse, I mean, I think 1 million's like the lower end. They recommend up to 4. So that's not always easy to get. We've been a little worried about that too. I mean, like regular PBMC data, I think has been fine. We've done some other tumor samples where there's some tumor and stromal cell types there, and we've had a couple experiments where the representation of cell types that we get is not really what's expected based on the experiment. And after we had done the ABRF study, looking at parse and other methods for cryopreserving total human leukocytes, which had granulocytes and everything in there, and we broke the kit by including granulocytes. Clearly, there are some cell types that shouldn't be in there that can cause issues. So I'm a little bit skeptical about, again, just certain types of cells, being overrepresented in the data at the end, just because how they're impacted by the fixation and the process. So yeah, that's a concern. But honeycomb is great. I don't know. It's a little weird. You sort of do, I don't know, some of like the library cleanup is on these like filter plates on a vacuum manifold. Like there's some things that are weird, but they give you all the stuff you need. And like once you've done it once or twice, it's pretty straightforward. The other thing we like it for is for like biofluids, if anybody's doing that. So we have some folks working with cerebral spinal fluid. We have folks doing urine, and a few other things where you have a lot of volume but not a lot of cells. And so you can actually put like a couple mils of fluid into the device, centrifuge it to get your cells to settle, and then if you still need more cells, you can actually remove that couple mils, add on another couple mils, spin again. And so that's also nice. Or even if people have like flow sorted, but you guys probably get this for 10x, right? Like people show up with like, I have 10,000 cells, but it's in a mil. It's like, well, okay, we're gonna lose half of those when we spin them and get them to
is
the
one
right
of the most
concentration.
frustrating things, especially I deal with it all the time because a lot of the PIs that come to me are junior investigators and they've never done this ever before. And I will send them the beautiful little chart that 10x does and like highlight, be like, I want this cell concentration for your cell type. And this is the quantity that I need you to give me because I can't load anymore. Like it's not an option. There's a limit to how much I can load. And I just dealt with that probably 3 weeks ago, that the PI then refused to let me spin her samples down and just pull off a little bit of extra liquid to get it to the concentration she needed for how much she wanted to target, refused to let me do that, and then was upset that I couldn't target the amount of cells that we had talked about. And I was
only
like,
so much you can do.
I was like, I don't think you understand. Like, I have to do this. She was like, well, you won't be able to see the pellet. There's not that many cells. I was like, I don't need to see the pellet. I just need to spin it down to I know that there should be a pellet right there. And I will just pull off some liquid and then resuspend my pellet. And we will run with that. And she refused to do that. And then was mad.
We don't give that much choice. Like if it's not concentrated enough, we're going to concentrate it.
Yeah.
That's protocol, and that's what we're going to do.
Yeah. We routinely have people sort into, I think
Sorry.
people are just used to sorting into several hundred microliters or a mil, but yeah, we frequently are like, 10 microliters at the bottom of the tube. I mean, especially if you're sorting something rare, right? It's like 10 microliters at the bottom of the tube. And yeah, there's a lot of times where we're just taking like 2 microliters of that for QC, and then the rest goes on the controller, right? It's just like, okay, make sure we're loading something and it's roughly where we want it to be, but it's a lot easier to dilute than to spin and concentrate.
Yes. Yeah, she also gave some of it to me in like 2 mils. She gave me like 4 samples. 2 of them were in 2 mils. And then the other one
out again.
Other 2 were in 1 mil. And I was like, really? Why can't you be consistent at least on all 4 samples? All right.
I think this is helpful. I think this is something we need to do more frequently because we could talk all day.
Totally. Yeah,
about all of our
absolutely.
issues.
No, this is very helpful.
Oh, so it's Kevin now, so we can't
Well,
complain.
Yeah, I guess 15 minutes wasn't enough additional time either, was it?
No,
That's okay.
I think this topic is passionate to so many of us that I think we could talk for weeks and still there wouldn't be enough time. So thank you all. And so let's kind of just go through the different topics and talk a little bit about. I'm just going to highlight. I was in the short reads session and highlight some of the key topics that were discussed in your sessions, and we'll go down the line. But really, in the short reads session, it was all about the money. Folks were why are we looking at going? We have an Illumina system, we have a NovaSeq or some. We want to have sequence our smaller projects at the same cost that it costs to feed the higher throughput Illumina systems. And so that's really one of the major reasons why folks are looking at that, looking there at the. There was some discussion on working with low diversity libraries. Didn't seem to be an issue with the element. Some are going with, instead of doing the 5% recommended, 10% is what they're going with instead. Then also the same way when you're spiking in phi-x for bisulfite conversion. So hopefully I've covered the major highlights, and we can move on to the next topic. That would be the spatial omics. Spatial.
Hello. Yeah, quick report back. So there is time for discussion. First of all, the number of people and the heated discussion showed exactly